My name is Tiffany Advincula, and I will be presenting on the duck-billed platypus. Platypuses live in freshwater rivers, streams, and lakes in eastern and southeastern Australia. They are generally solitary animals but can share a body of water with other platypuses. There is no evidence that platypuses communicate with each other in the wild, though they have been recorded to make growling noises in captivity. They are polygamous animals and can mate with more than one individual during any given mating season that occurs during the Australian winter. The most important part of a platypus's self-world is their bill. The platypus bill is an incredible sensory organ covered with sensitive receptors called mechanoreceptors. These receptors allow the platypus to detect electrical signals generated by the movement of the prey in the water, basically being its guide in this murky environment we find them in. Another important aspect is that their insulated fur helps them stay dry on a deeper level and protects them from the temperature of their environment. The auditory system assists them in their perception of the electromechanical feedback response to the structure of their cochlea. Platypuses exhibit positive chemotaxis when they respond to the chemical electrical stimuli given off by the smaller prey they hunt. Platypuses exposed to stress upregulate genes related to cellular homeostasis, metal detoxification, and antioxidation. For example, in platypuses from Single Lake, which had poorer water quality, these genes were upregulated. Exposure to heavy metals can induce oxidative stress in platypuses by increasing reactive oxygen species levels and decreasing catalase content. Overall, exposure to high levels of heavy metal can be harmful or even lethal. After hatching from eggs, young platypuses are highly dependent on their mother's milk and need to be in a safe environment to grow and develop properly. This period is crucial for their socialization and learning behaviors essential for survival. Any disruptions during this time can significantly impact their development and future success in the wild. For my question, I am asking, how does the availability of different types of food sources affect the foraging behavior and energy expenditure of platypuses? Platypuses will spend significantly more time foraging and demonstrate higher energy expenditure in these environments rich in their preferred food sources like aquatic invertebrates compared to the environments where these sources are scarce. This touches upon the functional aspect of Tinbergen's 4 questions. Since continuous sampling is best for monitoring detailed behavior over a period of time, I will record every occurrence of each behavior in real time during a defined observation period. This method allows for comprehensive understanding of how often and how long each behavior occurs. To enhance the data, I will incorporate the use of technology like cameras and drones. I will also account the time of day, weather conditions, and food availability to certify my results, and I will combine observational data with other methods like heart rate or dietary analysis. For my ethogram, I will be looking at foraging, diving, bubbling, swimming, and resting. This video is a good example of what we will be looking at in our ethogram. And these are my sources.